Hi, my name is Eric Lyons, and I'm a professor at the University of Arizona. This video is for my niece, Natalie Michaels, and her 5th grade science class. Natalie asked me to put together a short video about cell and DNA biology because she knows that I study genome biology here at the university. However, before I dive into those topics, I want to make sure people remember the wonder of what it is that we're studying when we do study the natural sciences. One of the things that I've always loved about biology is the sheer number of stories that there are to tell about the natural world around us. And one of the things that I love more than that is that the stories to be told really depend upon the person who's telling them. So, for example, if you take a look at this picture, a botanist may focus in on these white flowers and be able to tell us a story about their natural history. So that botanist may say, if you were to bend down and smell these white flowers, I bet they would be sickly sweet, characteristic of flowers that are pollinated by flies and bees. And look, there's a fly. Well, now an entomologist, somebody who studies insects, may take a look at the exact same picture and say, you know what? That fly is a very special kind of fly. That is in the family Saccharofagicidae, or the common name of flesh fly. Those are the flies whose females fly around looking for dead, rotting, decomposing meat in which to lay their eggs so that they're developing larvae, or children have a delicious and nutritive environment in which to grow up and develop. However, it doesn't look like that fly is going to get too many chances to lay eggs because it's been nabbed by this spider. Well, now an arachnologist or somebody who studies spiders may come along and say, you know what? That is a very special kind of spider. That is in the family Thomisidae, whose common name is crab spider. These are classic ambush predators where these spiders wait on top of flowers for unsuspecting pollinators, flies or bees, to come along, and when they do, they can nab them, bite them, and inject them with some neurotoxins that paralyze them, as well as digestive enzymes that liquefy the inside of that insect so that the spider can slurp up its own delicious meal. Well, you may notice that this spider is slightly difficult to see because it is cryptically colored. Now, an ecologist may come along and say, you know what? That cryptic coloration is very interesting. It turns out that it's not cryptically colored to hide from prey, such as this fly. And in fact, these dark markings on its abdomen are highly reflective in UV light, which means that it is highly visible to flies and bees. And it turns out that flowers that have these spiders on them are visited much more frequently by pollinators than those without, and that the reason it's cryptically colored is to hide from predators that are interested in preying on the spider itself. And what I love about this picture is I didn't even see this spider when I took it until after I had a chance to come back to a computer and look at it on a large screen. Now, what's important to, of course, realize about biology is that the stories go on from there. We don't just live in this static world illustrated by this one picture. We have this wonderful thing known as behavior. So this is a video from a David Attenborough video series called Life of Birds. And what we're looking at here is a male bird of paradise doing its best to impress a female bird of paradise. And what we see here is that there is this beautiful morphology, beautiful behavior that this bird is exhibiting in order to try to attract a female. And what's great about biology is that the stories don't end at the macro world, they extend down into the micro world. And so what we're looking at here is a beautiful animation done by Harvard's BioVisions, watching a white blood cell, what we call a leukocyte, circulating in your circulatory system, identifying a site of inflammation or infection, and then triggering a whole bunch of intracellular responses so that this white blood cell can react to this potential site of infection. And so what happens at a molecular scale is that the cell needs to communicate information that it is detecting from the outside inside of the cell. And so what this animation does is it brings together tens, 20s, 30 years of work in molecular biology done by hundreds and thousands of scientists that have spent a huge amount of energy trying to understand and characterize these very dynamic intracellular processes. Now, when I was in school, I was taught more or less that cells are these bags of goo, and instead what they are these highly structured and dynamic machines. For example, here's one of my favorite proteins, kinesin, that's walking along the intracellular cytoskeleton or highway, moving vesicles or large bags from one part of a cell to another part of a cell. Here we're taking a look at the nucleus, where out of the nucleus are coming messenger RNAs. These messenger RNAs are information that is transcribed from the genome or the DNA of an organism, and then that information is translated into proteins, the effective units that act within a cell to give it biochemical function and biochemical structures. Here we're taking a look at the process of ribosomes attaching to mRNAs and then translating that information into proteins that then get packed into vesicles. So this entire system, called the endoplasmic reticulum and the Golgi apparatus, are responsible for taking stuff from inside of the cell to outside of the cell. So here what we see are proteins that are going to be emerging on the cell surface of this white blood cell, and when they go ahead and get activated, they're going to change their shape and be able to now respond to what's happening outside of the cell. And so this is very important. Cells are these highly dynamic molecular machines, albeit a little wet and squishy, but they perform these incredible functions. In this case, we can watch this white blood cell flatten and then slip out of the circulatory system between two epithelial cells into the surrounding tissue in order to address the site of inflammation. So now the part that I find absolutely fascinating, where most of my research is focused, is trying to understand the structure, dynamics, and evolution of genomes. So the genome of an organism comprises of all of the DNA that makes an organism whatever type of organism it is. So our genomes, for example, are broken up into 23 pairs of chromosomes, half of which we get from mom and half of which we get from dad. And as you're probably learning, DNA is made up of 4 basic building blocks that we like to represent as A, T, C's, and G's. And so what a lot of my job is around is designing software in order to try to make sense of what all those A, T, C's, and G's mean. And the reason is that when we go out and we get the genome of an organism, we sequence the genome, this is how the information looks like, which is just a bunch of these A, T, C's, and G's that just go on and on and on and on. And very few people can take a look at this kind of information in this form and just start reading off and say, look, TGT, ATG, TTT, CAGG, AAA. I know exactly what organism that is. That is from the bacteria E. coli. So a lot of what I have to do with my job is try to come up with creative software systems that help scientists across the entire world make sense of all this DNA information. And so a lot of what I have to do is come up with these creative metrics and methods so people can understand what these data are and what they mean. So one of the things that I want to do is I want to introduce to you guys a brand new term that I'm going to invent called the Harry Potter equivalence. So I hope some of you have heard of the books Harry Potter, but if you have read them and have read all 7 novels, give yourself a big round of applause, because those 7 novels, if you were to add them all together, span about 1,400 pages. In those pages are about 560,000 words that are made up of about 3 million individual characters, the individual letters and spaces that make up words. If we take a look at genomes and try to understand the amount of information that we have to deal with when it comes to genomes, I want to relate them to what I'm going to call this Harry Potter equivalence. So the number of Harry Potters of those 7 novels, if you stick them all together into one book. So if we take a look at that bacteria, E. coli, its genome size is about 4.5, 4.6 million nucleotides or million bases long. It comes out to about 1 to 1 1/3 of these Harry Potter equivalences, or HPEs. If we take a look at more complicated organisms like yeast, which makes bread, its genome size is bigger. It's about 12 to 12.5 million nucleotides, or about 4 HPEs. If we take a look at some more complex organisms, and these are what we call model systems that we use in science to try to understand how plants work or animals work. All of these 3 here are commonly used. We have one that's known as Arabidopsis thaliana, our model plant. We've got Drosophila melanogaster, the fruit fly, which represents one type of animal, and then C. elegans, the elegant roundworm. All of these things have a genome size at the lower end of multicellular organisms, about 100 to 120 million nucleotides. That comes out to about 37 HPEs. If we take a look at humans, our human genome is often estimated to be about 3 billion nucleotides long, or over 1,000 Harry Potter equivalences. That's a lot of Harry Potter to read. And to put that kind of a number in perspective, if you were to read Harry Potter's for 8 hours a day, reading at about 1 page per minute, and reading these day after day, with no holidays, no vacation, no weekends, no sick days, 8 hours a day, day after day after day, it would take you about 8.25 years to read 1,000 Harry Potters, all 7 novels. And what I want to emphasize is this only represents 1/2 of a human genome. This would be the set of chromosomes from mom or dad, and that we have to double it if we want to really think about our entire genome. And what I want to emphasize is that genomes, the amount of DNA that go into making an organism, can vary greatly. So these are just some plant genomes. So Arabidopsis, the model plant used in biology, is really small for a plant, 37 HPEs. Some of the common plant genomes that are important for feeding us, like rice, maize, and wheat, these range from 100 HPEs to 6,000 HPEs, 6 times the size of the human genome. But when we take a look at the largest plant genome ever identified, which is a lily known as Paris japonica, it comes in at 50,000 HPEs. And what's important to realize is that as we're uncoding the language of DNA, and as scientists we're just beginning to have a feeling for where the words are and how those words might be used to construct sentences, and we have a long ways to go, is that we can't just use one single genome, we have to use many genomes in order to try to identify the areas of those genomes that are common and those that are different. And so, for example, if you take a look at these different kinds of corn, maize is another word for corn, the genetic difference between 2 of these different kinds of corn is about the same as what we find between humans and chimpanzees. So a lot of genetic, what we call diversity, differences can occur, and that those differences really can make these things look very different or behave very differently. So in some regards, humans and chimps are very similar. We look overall the same body plan. We're both primates, but we have a lot of differences, especially when it comes to behavior and how we think and how we learn. So this comes down to my job, which is to help people transform genomes of information into knowledge. And the thing that's really important to realize is that this transformative process, changing information into knowledge, only happens in one place, and that is in the trained brain of scientists. And what we do in my group is we come up with a variety of computer software and data management systems to help convert all of that information into some form that the human brain can process more easily, like images or interactive graphics that then can be fed back in so a biologist can understand. And if you take a look at this image, and this image represents this genomic region, but instead of being A, T, C's, and G's, this graphic represents genes, the regions of genome that code for protein, by these colored arrows. And a scientist may look at this and say, look at that. This gene right here labeled IS5, which is a special kind of gene, has landed here in this genome and has broken up this other gene into two pieces. This gene called IS5 belongs to a class of genes that make copies of themselves and then jump to a new location of the genome and insert themselves in there. So what's happened is that this gene has jumped to a new location, broken the DNA there, and inserted itself to make a copy. But in the process, it damaged and made this other gene non-functional. So what the biologist can now do is try to figure out how this may have affected the overall functioning of this bacteria as a whole. So as you go forth and you learn about science, and biology in particular, just remember that there are many stories to tell, and there's many stories left to be told. And all it takes is a little bit of curiosity and usually a lot of patience just to go out there and look to see what's happening, and then to be able to put that together in a way that allows you to transform all of that natural information into useful knowledge so that you can go out there and do good things. So what this is part of a bigger process that's happening across science called data-driven discovery, where our ability to observe the world around us is changing rapidly as new technology becomes available that lets us record what's happening at an unprecedented scale, requiring a lot of information, a lot of data. And if you have any questions, please feel free to contact me. My name again is Eric Lyons. I'm at the University of Arizona. I'm in the College of Agriculture and Life Sciences. I'm part of the School of Plant Sciences. I'm part of a large project to build software to help scientists make discoveries called iPlant. I'm also a member of the Bio5 Institute, and I'm part of the Graduate Interdisciplinary Program in Genetics. Please feel free to contact me at elyons.uoa@gmail.com.